Hello friends, I am Lakshmikant. Hope you all are fine, my dear. Here in this video, I am going to start Unit 9 Biotechnology Principles and Process, my dear. Here in this very topic, this one is very important chapter, my dear, at the point of what NEET examination, my dear. So here I am going to start these all things. Before we have to start, let's see that what we should have to study here, my dear. Here in this very topic, biotechnology, in two way we have to be study here. The first, the principles and process, then after we study about the application, my dear, right? So first we see the principles and their processes, then after in next chapter we start what applications of this one chapter, my dear. Okay, so unit 9 biotechnology, class 12. Let's see what we should have to know here. Here, dear, first of all, I'm going to tell you about the term that is what biotechnology. You know that the meaning of bio, yes, sir. Bio means what? Life. Let's see what I'm going to tell you here. Let's see first. I'm going to write here the term word biotechnology. See, sorry, this one is the term biotechnology. Getting technology, my dear. Wait for a moment. Suppose if I'm writing here the term biotechnology, you know what the meaning of this one thing, my dear? Biotechnology. Here, the biotechnology, it means what? Let's see first this one thing. First, the term here is what? Bio. You all know that bio means what? Bio means what, my dear? You know that bio means what? Life. What I'm saying? Bio means what? Life. Getting? And here the term technology, here the term that is what, my dear? Technology. I'm just separating this one thing, technology. Here what the technique that we have to be study in biotechnology, my dear. So see, if I'm saying here that biotechnology is a technique, what I'm saying, it is a technique that how the natural science and organisms, cells, parts, and molecular analogues for products and services, my dear. Getting my point? So see, if I'm saying here that the process of the rDNA technology is how we cut the DNA and how we study the specific location of restriction enzymes, my dear. Getting my point? So how we isolate the genetic materials, these all things where we study, these all things we study in biotechnology, my dears. What the principles of genetic engineering where we study in the biotechnology principles and processes, my dear. Getting my point? So see, if I'm saying here that the biotechnology is such a tools that we study each and everything whatever we want to change in their life, my dear. Whatever we want to any modification in their life, we must have a word knowledge of what biotechnology, and we hope so that this technology is applied in various fields, my dear, in medicines, in agricultures, in you say every places that we need to generate something new, their biotechnology use, my dear. So see, what I'm saying to you in this very mind map, I'm going to tell you about this biotechnology. Let's see with the help of the word, the things that is given in NCERT also, my dear. See here in NCERT, this one is the unit 9 biotechnology, my dear, right? Getting my point? So if I'm saying here that this one is the biotechnologies, and here the chapter number 11 biotechnology principles and what processes, my dear. Getting? See what I'm saying here. I'm saying here, dear, that if I'm studying this one chapter, so what the main motive to understand here? Here the main motive to stand here the biotechnology. What do you mean by biotechnology? This one is the question, right? So if I'm saying here the biotechnology that deals with the technique of using live organisms, what I'm saying to you, live organisms, that organisms which is alive, or you say what enzymes from organisms to produce products and processes useful to human, my dear. Getting my point. I'm saying here that biotechnology deals with techniques. What I'm saying? Deals with what techniques? And this one techniques using live organisms, my dear. What I'm saying to you? Live organisms. And that with the live organisms or enzymes from organism to produce products. And what processes that is very useful for mankind, my dear. Getting my point? So let's see if I'm saying this one thing, it means that here making curd, bread, wine, whatever the things which are all microbe mediated process would also be thought to form from which one? Biotechnology. So what a fantastic thing here, my dear, that whenever we want to study this one thing, that is important to understand. It means that in each and everyday practical life there must be a word biotechnology, isn't it or not? So see, it is, see what the NCERT is written here, it is used in a what restricted sense today, definitely, to refer to such of those processes which use genetically modified organisms. What I'm saying, GMO, I'm saying about this GMO. This GMO means what? Genetically modified organisms. And how it be possible? So it can only be possible with the help of biotechnology. So see, if the GMO to achieve the same on the larger scale, so we should have to be modified now. But how this one? How this one? Let's see. Many other processes and techniques are also used under biotechnology. You know what? Let's see some examples. In vitro fertilization, IVF. Suppose if the couples not leads to birth to their young ones, then that time it means that that couple is what? Infertile. I'm not saying sterile. I'm saying what? Infertile. If the marriage will be take place and after 2 to 3 years they doesn't give birth to their young ones, then what do you mean? It means that they are sterile? No, my dear, that is infertile. Because the sterility and infertility is two things that is totally different from each other. Getting my point or not? As sterility, it means that the permanent, they do not give birth to their young ones, then it is called a sterility. But if I'm saying here that infertility, it means that they have chances to give birth to their young ones, but the condition is be that they not have the matured sperms or motile sperms, either from the male side defect also, either from the female side also defect. That very cases, is it possible to give to their birth of young ones, my dear? Yes, definitely. There is a technique. That technique is known as IVF, in vitro fertilization. The second one, again, I'm saying IVF, that is called in vivo fertilization. In vitro fertilization, that is the fertilization outside the body cells. And if I'm saying the in vivo fertilization, it means that fertilization within the body cells. Let we discuss these all very things, but first of all here, and this one hope so, this in vitro and vivo fertilization is given in reproductive health. Yes, today I will start the live classes on reproductive health, my dear, at 9 p.m. You must join. Okay, so in that very cases we study about these all things, isn't it or not? That is the last paragraph that is given in vitro and vivo fertilization. Yes, that is human reproductive health, my dear, right? Yes, let's see. Here, in vitro fertilization leading to what? Test tube baby. What I'm saying? Test tube baby. So in this very synthesizing a gene and using a developing DNA vaccine or correcting a defective gene. Suppose if any the gene is very defective, then it is possible to be become to correct. Yes, sir. Definitely. So what way, how we correct these all variants if the gene is were defective? If the DNA vac, yeah, that is a DNA vaccine. This is the latest technology that is used by the scientists to cure the any diseases, my dear. Let's let's see how how this one is response. How this one is what possible, my dear. See, the European Federation Biotechnology. See this one is the line EFP. That is what? Sorry, EFB. That is European Federation Biotechnology has a given a definition of biotechnology. You know what? You know what? That encompass both traditional view and modern molecular biotechnology. The definition given by EFB is what? This one is very important, my dear. See what the definition given by the EFB, European Federation of Biotechnology. According to EFB, the integration of natural science and organisms, cells, parts therefore, and molecular analogous for products and services. Getting, getting. So this is known as what, my dear? Biotechnology. So see, the integration of natural science and organisms. So if I'm saying here the integration of natural science and organisms, parts thereof, the molecular analogous for products and services, my dear, then it is known as what? Biotechnology. So this one is the definition, which is very, very important to understand, my dear. So write in your copy, mark in your NCERT. This one is very, very important. Hope so you understand this term, biotechnology. Definitely. The integration of natural science. Suppose if I'm saying here that if the science, natural science to be integrated, where? In the organisms, in the parts of and the molecular analogous, in the products and services is known as what? Now the thing that is important to understand, sir, what the thing that is important to understand, let's see, my dear, what the things that is very important to understand here, the some principles of biotechnology. You know the principles of biotechnology. Let's see one by one that was the thing is given, what the thing is given in NCERT. You know, my dear, NCERT lines are very, very important. You go through line by line, you go through word to word, then one day achieve your goal, my dear. So if you not go through line by line or word to word from NCERT, then you can't achieve your goal, my dear. That is your goal, that to crack the NEET. Am I right? If you want to crack the NEET, my dear, then you have to be go through line by line or word to word in NCERT, my dear. Right? Let's see. Among many two core technique. What is saying? Two core technique that enable the birth of modern biotechnology. So what the two core technique? You know, first one is what? Genetic engineering, my dear. What is saying? Genetic engineering. You know that so many people want to do genetic engineering. You know why? Because first, before why the people go in genetic engineering, this one is not a matter. Let's see first that what is genetic engineering. Getting? So genetic engineering is what? Genetic engineering is what? Technique to alter the chemistry of genetic material. So if anyone know the technique to alter means what? To change, alter the chemistry of genetic material. That is what? DNA and RNA. You know that genetic material is what? DNA and RNA. DNA content where on a chromosomes? How much percent DNA content on chromosomes, my dear? Yes, this one is the question for you. I'm asking. Yes, I hope you know this one answer. Only 40%, my dear. 40% DNA content on chromosomes. Rest 60% that is containing what? You know, rest 60% containing histone protein, my dear. Correct? So if anybody can ask you that what is genetic engineering, then what do you say? In that time you say here the technique to alter the chemistry of genetic material. If I'm saying here that the alter the chemistry of genetic material, my dear, then it's known as what? Genetic engineering. And this one to introduce this into host organism. Yes. How it be possible, sir? It can be only possible whenever the this genetic material introduced into here host body cell. Hope so you know the meaning of host. Yes, that is a protective covering body, my dear. Whenever I'm saying this one, the host body cells. Yes, host body cells, because once the any pathogens invade within the body cells, they are first transmitted through host body cells, my dear. So here also the technique to introduce the word genetic material into their host organisms and thus changes the phenotype of the host organisms. Phenotype, you know the phenotype, genotype. Yes, my dear, principles of inheritance chapter. Yes, phenotype, that is features that can be seen outside from the, yes or no? Outside, if I'm looking smart, so this one is the phenotypic ratio. Yes, if anyone looking dark, or anyone looking just dwarf, or anyone looking just tall, that is gigantism, then that is a what? Phenotypic. Yes or no? So whatever the observable features we see from outside, that is known as what? Phenotypic, right? Again, I'm repeating, whatever the observable feature from outside, getting my point? Whatever the observable features observing from outside, then it is called what? Definitely right. Very good. That is what? Phenotypic. Let's see another thing in a genetic engineering. What? Maintenance of sterile. Wow, this one is so important. Yes, in a genetic engineering, here the way that we have to maintain the sterile and just mean period of time. I'm just telling you about this one thing, that is what sterile. Sterile means what? Sterile means that the couples, which is what infert, and the couples were not able to reproduce their young ones. So that couple is said to be what? A sterile. So maintenance of a sterile, that is what? Microbial contamination free. Yes, so ambience in a chemical engineering process to enable growth of only the desired microbe. If I'm if I want to do with what grow a microbe, then there is a what desired microbe. Eukaryotic cell in a large quantities for the manufacture of biotechnology products, my dear, like antibiotics, vaccine, enzyme. Suppose if anyone have a diseases and that very disease is not cured, then that time the doctor will give what? Antibiotics. This one, antibiotics, enzymes, vaccine, these all are the things which is manufactured with the help of what? Biotechnology, my dear. So without biotechnology, imagine how the antibiotics, how the vaccine, how the enzymes prepared, my dear. Getting my point or not? So see, this one, biotechnology is very vital, or you say the very vital role play in the developing the what you say the mankind, whatever the basic things required by the mankind, that all the things is fulfilled by the help of what? Which one? That is biotechnology, my dear. See, next thing that is important to understand here, you probably appreciate the advantage, you know, advantage of what? Sexual reproduction over asexual reproduction. Why I'm saying this one line, because sexual reproduction there is a variation, while asexual reproduction doesn't containing variation, my dear. And if the variation is not be there, then the evolution is also not be there, because I was told you in previous lecture that is variation is the key factor of evolution, my dear. So that's why I'm saying here that the probably we appreciate the advantage of what? Sexual reproduction over asexual reproduction. Here the farmers provides opportunity for variation. See, this one line is given in NCERT. And formulation of unique combination of genetic setup. Getting my point? Getting my point? So asexual reproduction preserve the genetic information, whatever the in the genetic information they preserve. While I'm saying sexual reproduction, my dear, permits variation. So this one is the differences between sexual and asexual reproductions. Yes or no? So traditional hybridization. What's the meaning of hybridization, sir? Hybridization means that the technique in which the desired characters can be formed by crossing over the undesired ones. Getting my point? So traditional hybridization procedures used in plants and animals to breed very often lead to the inclusions and multiplication of undesirable genes. See what I'm saying here? If I want to change the undesirable gene along with the desired gene, then what we should have to do? We should have to use what, my dear? A technique. That very technique is known as what? Hybridization. So dear, hybridization is also a technique in which procedures to use the plants and animals breeding very often including of multiplication undesirable gene with desirable gene, or you say the desired gene. Here the technique of genetic engineering. This one is the technique, genetic engineering, which include creation of what? Recombinant DNA. What I'm saying? Recombinant DNA. Recombinant DNA, it means that the DNA will what? Change in their features. Use of genetic cloning, gene transfer, these all are the process, these all are the way that how undesirable genes along with the word desired genes by dears to be transmit. So see, overcome this limitation, allow in to isolate and introduce only one or set of desirable gene without introducing undesirable gene into the target organism. Sir, is this one possible? Yes, my dear. This one only possible whenever we transmitting the genetic material from parents to their what? Progeny, my dear. Getting my point? Do you know the likely to fit to piece of DNA? Yes, first of all, we should have to take the piece of the DNA which is somehow transferred into alien organisms. If I'm saying the alien organisms, it means that the organisms that is what? Totally different. So mostly like the pieces of DNA would not be able to multiply itself. Yes, sir, definitely DNA would not multiply itself. The progeny cell in the but when it gets integrated into the genome, what is saying? Genome of the recipient, it may multiply and inherited along with the host DNA. So if it is introduced, then the progeny cells integrated into genome of recipient, and which when is responsible to multiply this inherited along with the host of DNA material. Correct? Connect this in this way we have to be studied. So this is because the aligned pieces of DNA has become a part of chromosomes, which has ability to what replicate, or you see the chains. So replication of the DNA, it means what they are making the copy of what DNA, my dear. So in a chromosomes there is a specific DNA sequence. Yes sir, definitely that very DNA sequence, the chromosomes, there is a specific DNA sequence called what, origin of replication. So see what I'm saying here that suppose this one is what, DNA, and here I'm representing O. So here O is the what, origin of replication. From here the enzymes which is responsible to replicate this one. You know, this one is what, replication of enzymes, my dear. See, in a chromosomes there is a specific DNA sequence called origin of replication, and this origin of replication is responsible for initiating what, replication. So if anybody can ask you from where the DNA replication, so it replication at the point, that name of that very point is called origin of replication. Getting? So therefore, for the multiplication of any aligned pieces of DNA in an organisms, it needs to be a part of chromosomes. It needs to be what? Part of chromosomes. Without the parts of chromosomes, which has a specific sequence known as origin of replication, then we can't imagine that the replication of DNA will take place. So thus, an aligned DNA is linked with the origin of replication, so that the pieces of aligned pieces of DNA can replicate and multiply itself in the host organisms, my dear. Getting my point? Yes. So this can also be called cloning, or you say the what? Making multiple identical copies of any templates of what? DNA, deoxyribonucleic acid. See, what I'm saying here. I'm saying here that the aligned DNA is linked. When I'm saying that aligned DNA is linked, aligned DNA is linked with the origin of replication. Yes, and this aligned DNA can replicate and multiply itself in the host organisms also. And this very process is known as what, cloning. And this one cloning will be only performed whenever the making multiple identical copies, my dear, of any templates, that is what DNA. Let us now focus where, on the first instance of the construction of artificial recombinant DNA molecule. Getting? So if I'm saying here that the recombinant DNA molecules, so this recombinant DNA molecule is responsible for the making a what? A what? A copies, my dear. See, the construction of the first recombinant DNA emerged from the possibility of linkage a gene. What I'm saying? Linkage a gene. That a linkage a gene encoding antibiotic resistance with a native plasmid, my dear. So see, if I'm saying here that the native plasmid, which is responsible for making this first recombinant DNA known as what, autonomously, what I'm saying here, autonomously replicating circular extra chromosomal DNA. So see, if I'm saying here that the extra chromosomal DNA, which is responsible for the transmitting of the genetic materials from one of the parents to their other host body cells, my dear, let's see which one is the what a scientist who give, and which one is the scientists first we see here, that is Salmonella typhimum, Stanley Cohen, and Herbert Boyer, my dear, accomplished in these all things in 1972. And you know that this isolating the antibiotic resistant gene, what I'm saying, antibiotic resistant gene, and this antibiotic resistant gene is used by cutting the piece of our DNA from where? From a plasmid which was responsible for conferring the antibiotic resistance, my dear. Getting my point? Again, I'm going to repeat here. I'm going to repeat here. The Herbert Boyer, the scientist who isolate the antibiotic resistant gene. Okay, and then after what? Cutting out the piece of the DNA from a plasmid, and then after responsible to confront the antibiotic resistance. Now this cutting DNA at the specific location become possible only whenever the discovery of what enzymes, my dear, that also molecular seizures, or you say the restriction enzyme. How it possible to cut that the DNA, my dear? Yes, sir. With the help of what the enzymes, the name of the enzyme is called restriction enzyme. So whatever the cut piece of DNA was then linked with plasmid. Here it will link with the plasmid DNA, and then that very plasmid DNA act as a what vectors. So vector is what carriers of the what transfer piece of DNA attached to it. So where the where the DNA added, so plasmid DNA act as a vectors, my dear. So the cut pieces of DNA was then linked with the plasmid. So once the plasmid is linked with the cut DNA, this plasmid DNA act as a what, my dear, vectors, and this very vectors transfer the piece of a DNA to attach to it. You probably know the mosquito act as an insect vector, my dear. Yes, definitely, sir. So see, see what I'm saying here. To transfer the malarial parasite, what I'm saying, malarial parasite, my dear, into the human body. In the same way, plasmid can be used as a vector. Getting my point? So see, if I'm saying here the malarial parasite, whenever it transmits with the help of the what? Mosquito into human body, in the same way, the plasmid can use as a vector, my dear, to deliver an aligned piece of what? DNA. So see this one thing. So this one thing, I'm saying that the plasmid is used as a vector to deliver the aligned piece of DNA. Suppose this one is the organism. Just wait for a moment. I'm going to show you with the help of, suppose this one is what, this one is what, this one is aligned. What I'm saying to you here, I'm saying what, aligned. Clear? So see, if I'm saying here the plasmid can be introduced as well as a vector to deliver aligned pieces of DNA, where? Into the host of another organisms, my dear. Getting my point? Getting my point? See, so the linkage of antibiotic resistant gene. What is saying? What the linkage? Antibiotic resistant gene with a plasmid vectors become possible with an enzyme DNA ligase. So in that time, the DNA ligase is responsible to act on cut DNA. Where it act? It act where? On a cut DNA molecules and join their ends, my dear. So DNA ligase is an enzyme which is used to what, join the cut DNA, while the restriction enzyme is responsible to cut the what DNA. Getting? So this makes a new combination of what, circular autonomously replicating DNA created in vitro and is known as what, recombinant DNA, my dear. What I'm saying here? Recombinant DNA. So this one recombinant DNA technologies which is responsible for carrying the what, DNA from desired ones to be what, undesirable. So when this DNA is transferred into the what, E. coli. See what I'm saying? E. coli. Clear? A bacterium closely related to a salmonella. What I'm saying? What I'm saying? A bacterium. That very bacterium that is what closely related to salmonella. It could replicate using the new host DNA polymerase enzymes. See what I'm saying? So if they replicate using the new host DNA polymerase enzyme, that very DNA polymerase enzyme is responsible to make what, multiple copies, my dear. What I'm saying? Multiple copies. So if this one multiple copies is going to made, my dear, so this one is only the possible with the help of the what, enzymes, my dear, that is DNA polymerase enzymes, my dear. So now see, the ability to multiply copies of antibiotic resistant gene, my dear. What I'm saying here, what I'm saying here, the ability to multiply copies of antibiotic resistant gene, where? in E. coli, my dear. E. coli bacteria we use, called what, my dear? Cloning. What I'm saying? Cloning. And this cloning of what? Antibiotic resistant gene. And this antibiotic resistant gene where, my dear? In E. coli bacteria. What I'm saying you? E. coli bacteria, my dear. So if anybody can ask you which one bacteria is used for cloning purposes, then what you say? So E. coli bacteria. Why I'm saying this one thing? Because E. coli bacteria called what? Antibiotic resistant gene, my dear. That is E. coli. So that's why we use this one thing, right? So now you can hence infer that there are three basic steps in genetically modified organisms. You know what are the basic things, my dear? Here the genetically modified organisms. Here are the three basic things that is very important to understand here. Number one, that is identification of DNA with what? Desirable gene. What I'm saying? Desirable gene. So see, here whenever I'm saying that this identification of DNA with a desirable gene, getting my point? Getting my point? So if I'm saying here that if that the first we should have to what? Identify the DNA. What is saying you? Identify the DNA, and this identification of DNA, that is a desirable genes, then after we what? Introduce this identified DNA where? Into the host body cells, my dear. After that, the maintenance of introduced DNA. What I'm saying? Introduce DNA in the host and transfer of this DNA to its what? Progeny, my dear. Getting getting this one thing. The first, that is what? Identification of DNA. Yes. Then after what? Introduction of the identified DNA into the host. Then third one is what, my dear? This maintenance of introduced DNA into the host. And finally, this transfer of the DNA into where? Progeny. So see, all these very techniques, all very these things, all with these various steps in a genetically modified organism, GMO, these all very steps. But the thing is important to understand here, sir, how it possible. Is there any tools? Yes, sir. That very tools is known as what? Tools of recombinant DNA technology, my dear. So let's see what the tools of recombinant DNA technology. Getting my point? Here I'm going to only show you the line of NCERT. When I'm going to teach you in a details video, my dear, then I will tell you with the help of the what, making the diagram, that how this tools, how this recombinant of DNA technology will take place, my dear. Let's see first here what the NCERT is telling you first. Here are the some tools. You know what the tools, my dear? Here are the tools. Here the tools of recombinant DNA technology. Let's see. Suppose if I am saying here that the foreign discussion, the genetic engineering, or you say the recombinant DNA technology, this can be accomplished only if we have to be key tools. So what the key tools? Number one, that is restriction enzyme. Yes. Second, polymerase enzyme. Third, ligase. Fourth, vectors. And other one is what? Host organisms. So these all are the tools, so that we see here that this one is the thing that is important to understand. The first thing, let's see, my dear, in the first thing, that is what? Restriction enzyme I'm saying. What I'm saying? Restriction enzyme. So see, in the year 1963, my dear, what I'm saying to you, 1963, the two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. You know, E. coli bacteria. Yes, in the year 1963, the two enzymes is responsible here for restricting the growth of bacteriophage. Where? In a E. coli. Let's see. One of these added a methyl group to DNA, while other cut DNA, my dear. So this one latter called known as restriction endonuclease. Again, I am repeating here. I am saying here that in the year 1963, the two enzyme is responsible for restricting the growth of bacteriophage in E. coli. You know, that is number one, that is what? Methylated group to DNA, while other is what? Cut DNA. So methylated group of DNA and the cut DNA were used. That is also known as what? Letter, that is also known as what? Restriction endonuclease enzymes, my dear. Getting? So thus first endonuclease, the first endonuclease, This one is very, very important to know. That is Hind2, my dear. What are you saying? This one is important line. So mark in your NCERT, my dear. You are not marking in your NCERT. Mark, mark here. This one thing. This one thing is important. Getting? Yes. Whose functioning dependent on a specific DNA nucleotide. What I'm saying here that whatever the restriction endonuclease, that is Hind2, which is responsible for using on a specific DNA nucleotide sequence which was isolated and characterized 5 years later, my dear. So it was found that Hind2 always cut DNA molecules at a particular point. What I'm saying here? What I'm saying here that if we use a restriction enzymes, that is restriction endonuclease Hind2, this Hind2 is always responsible to cut the DNA at a very particular point by recognizing a specific sequence of 6 base pair, 6 base pair, my dear. So see, this specific base sequence is known as the what? Recognition sequence, my dear. So where the recognition sequence, that where the restriction enzymes cut, that is restriction endonuclease enzymes Hind2. Besides Hind2, today we know more than 900 restriction enzyme, my dear. Yes, definitely. We know the 900 plus 900 restriction enzyme that have been isolated from 213 strains of bacteria, but in spite what we use in the two restriction enzymes here we use, my dear. Now, the next thing that is important to understand here, that the convention of naming these enzymes. So how these enzymes will be the name, my dear. The first letter of the name come from the genus and second come from the two letters, my dear. The first one, that is what Hind, 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 that is what genus come from the species of prokaryotic cells which are isolated. And E. coli comes from what? Echinacea coli R113. In E. coli, the letter R is derived from the name of what? Which one? Name of a strain. And Roman numbers following the name indicate the order in such the enzymes were isolated from the strain of bacteria, my dear. So let's see this how will is possible, sir. How this restriction enzyme is possible to make a process in a large scales. You know that this restriction enzymes belong to a larger class of enzymes, my dear, called nuclease. You know there are two kinds. One is the exonuclease. Second one is what? Endonuclease. This exonuclease or endonuclease, what it means, sir? Exonuclease means that if we remove the nucleotides from the end of DNA. What I'm saying? From the end of DNA. Suppose if I am making this one diagram, suppose this one is what? 5' end and this one is what? 3' end. Okay. So if I'm cutting this one DNA, let's see, this one is 3' end and this one is what? This one is what? 5' end. Okay. So suppose if I'm cut this one DNA, so here the some restriction endonuclease, the enzymes functioning of inspecting the length of the DNA sequence. So see, if I'm saying here that exonuclease, it means that exonuclease means that remove nucleotide from end of DNA, whereas endonuclease may cut a specific position within the DNA. So suppose if I am cutting any substances within the DNA, then it is known as what? Endonuclease. If I am saying here that cutting the specific position, not a specific position, my dear, we just remove the nucleotide from the ends of the DNA, then it is known as what? Exonuclease. Getting this one thing? Once it finds a specific organization, recognition sequence, it will bind to the DNA and cut with two strands of double helical structure specific point. That double helical structure specific point is the end of what? Sugar phosphate backbone, my dear. What I'm saying to you? Sugar phosphate backbones. And this sugar phosphate backbones is a word containing each restriction endonuclease recognize a specific sequence. That very specific sequence is known as palindromic nucleotide sequence in the DNA, my dear. This one is very, very important to understand. So any DNA there is endonuclease recognizes specific what? Palindromic nucleotide sequence. Here the enzymes cuts both DNA. See here, this one is guanine to cytosine having triple bond, adenine to thymine having double bond. See ATCG, ATCG. Here the two ends, that is guanine to cytosine, guanine to cytosine. Here the what I am saying that E. coli cuts the DNA between the basis of G, where it cut it from G and A, when the sequence GAATC is present in the DNA. Yes or no? So this one is the thing that is important to understand. But in details video, I will tell you about these all various steps in the formation of recombinant DNA by the action of what restriction endonuclease enzymes, my dear, that is E. coli. Okay. So in details video, whenever I tell you this one thing, then please show you, or I will check there that how this restriction enzyme is responsible to cut the DNA patterns, my dear. Okay, so thank you for watching. Thank you. Have a nice day. Take care. Bye bye. Just we meet, and soon we meet, my dear. Thank you.